The cause of plant, animal and human diseases had been a mystery since millennia. Also, it was not known that how disease transferred from one person to another person. So, number of theories were proposed to prove how diseases transferred from one organism to another organism. Number one is the miasma theory of disease. Miasma is a Greek word which means polluted air or bad air. So people believe that bad air was the cause of many diseases. And then came the theory of spontaneous generation. According to this theory, living matter originates from non-living matter spontaneously. So Francisco Redi in 1668 devised a set of experiments to disprove this theory of spontaneous generation. He actually took 3 flasks. Number one was unsealed, second was sealed, and third was covered with gauze. And it was noticed that maggots appeared on meat in unsealed flask, while there was no growth in sealed flask, and they appeared on gauze, in which the flask was covered with gauze. So it was enough to disprove the theory of spontaneous generation, but the proponents of theory of spontaneous generation were not satisfied. John Needham was one of the proponent of spontaneous generation theory. He devised a set of experiments to prove this theory. As we know that boiling kills microbial growth, he took chicken broth with microbial growth and he boiled it and then sealed it after boiling. Broth appeared clear of life after boiling, but after some days broth has microbes in it. So he tried to prove that the theory of spontaneous generation was right, but people who were against this theory, they argumented that it occurred because there was time duration between the boiling and the sealing. Lazzaro Spallanzani was against this theory. He redevised experiments of Needham to disprove this theory. He actually boiled the chicken broth and sealed it, and then he removed air from this flask, and he observed that there was no growth. But the proponents of this theory, they argued that air was also necessary for the growth. Then Louis Pasteur devised a set of experiments which finally disproved the spontaneous generation theory. What he did was he took non-sterile liquid into a flask, and then the neck of a flask was drawn out in S shape or swan neck shape with the help of this heat. And we can observe here that he then sterilized liquid by heating, and air was forced out. We can see here that dust and microbes can be trapped here. Air यहाँ से enter हो सकती है, लेकिन microbes due to this shape they cannot enter this liquid because of the gravity. After long time liquid remains sterile, but then what he did was कि उसने इनको flask को थोड़ा सा turn किया so that this liquid can come in contact with these microbes, and then we can see that microbes grow in liquid. So this theory finally disproved the theory of spontaneous generation. Louis Pasteur, in addition to this contribution, he also gave us the process of pasteurization or killing microbes from milk and giving it long shelf life. In addition, he discovered vaccines for various diseases. Then Joseph Lister, who was a surgeon, he did a very good job. He disinfected wounds after his surgeries, and he was first to devise this, and it limitized the deaths which were very common after surgery. Then the work of Robert Koch is very appreciable. He proved that a single microorganism is associated with a single disease, which Louis Pasteur could not prove. So he also gave the germ theory of disease, which have 4 postulates which are noteworthy. Number 1 is that the suspected germ must be present in every case of the disease. We can see here the disease subjects show the growth, while the healthy subject do not show the growth or do not show the presence of this organism or this specific germ. So the second postulate is that the germ must be isolated and grown in pure culture. कि हम उस disease में से उसी germ को isolate कर सकें in any case, उस single germ को. Number third postulate is that the culture germ must cause the disease when it is inoculated into a healthy, susceptible experimental host, whether it be a plant or animal. कि अगर हम उस germ को जो हमने इस pure culture में obtain किया है, इसको हम किसी healthy organism में inject करें, तो it may cause disease or it should cause disease. And number fourth postulate is that the same germ must be re-isolated from the disease experimental host. कि अगर उस organism को फिर से हम लोग उसमें observe करें और उसको check करें, तो उसमें से वो specific germ isolate होना चाहिए जो we consider is associated with the specific disease. So due to the contribution of these scientists, we have now come to know that a disease is related to a specific organism, or one disease is related to one specific organism. So due to the contribution of these scientists, we have now much more advancements in the field of medicine.